Hello, everybody. My name is Lam Mukherjee, and it is my pleasure to take you through the next 10 minutes where I summarize my team's progress and the plans for the future. We present to you the RECON series, a suite of solutions to microbiology and metagenomics that at its core champions the principles of early detection. Our focus diseases are neurological disorders, and our first candidate in the RECON series is the Alzheimer's disease. For today's agenda, I shall discuss the situation and a proposed solution for the problem in the project summary. Then we understand the possible roadblocks of the solution, both from a research perspective and a business one. Following this, the presentation covers methodology and discussions that we use to find our solution through our pipelines. We wrap it up with a business case and some concluding thoughts about what's next for the team and this project. We start with a simple case study. To set the stage, in Finland, with every one citizen, there are four who are above 80. The nation's population is predominantly of senior citizens. With the mortality rate of 52%, the problem is an existing one in the 21st century. Adding on to all these red flags, the cost per patient borders in the neighborhood of 24,000 euros. We can estimate that the annual commitment to such healthcare facilities can amount to upwards of 3,000 million euros. For a nation that boasts a relatively lower population and is considered developed, this might not be a concern, but it becomes one for developing nations to support their population in an age of customized and costly healthcare solutions. So let's consider the problem as it is. Taking its face value, we are aware of the rise in the overage population in developed countries. This skewed rise in senior population compared to the younger or the non-earning and the earning raises concerns about the nation's future. For developing countries, the ability to bear the increase in government spending for healthcare facilities is a problem, while existing tests for Alzheimer's disease are invasive and costly, and are most often than not provided by specialized centers. Additionally, some patients may also view them as invasive for the nature of these tests involve lumbar puncture or PET. And finally, the burden is unfairly laid on low to middle income countries as 60% of AD cases are reported in such nations. The solution exists. The solution exists in having the ability to aid individuals in being aware for the need of change. For Alzheimer's disease is often the result of habits and behaviors adopted by the individual over the course of their life. Early detection of microbial dysbiosis or any changes in the gut-brain microbiome axis can go a long way in helping individuals choose a better quality of life. Our solution is a microbiome testing kit that detects either the abundance or the absence of a specific bacterial species in the gut microbiome. In doing so, patients flagged with this dysbiosis can be introduced to diets of supplements and better health habits to potentially avert the potential problem. Sounds easy enough, doesn't it? Anyone could find a population of bacteria that will help anyone diagnose an individual with the possibility of developing Alzheimer's. Well, it isn't easy, and that is exactly what we understood during the project. But focusing on the positives, how will our solution work? Sample collection will involve collection of fecal samples from the individual. These samples will then be introduced to the testing cocktail, which, through chemical reactions, will introduce visual indicators, which will allow for a visual understanding of either the absence or the presence of microbial dysbiosis. All this is being done to make the platform easier for individuals to use. In the presence of dysbiosis, patients will be recommended to consume supplements and change their habits. Now, for the components of the EDK or the early detection kit account, according to us, will involve the analyte, the bacterial population that indicates the potential onset of Alzheimer's disease, the bioreceptor, which in this case is the cocktail, which will be a combination of enzymes and proteins. Excuse me. This, this cocktail will test positive or negative to the target microbial population. Finally, the interface of the platform will indicate the potential chances of being diagnosed with AD. We believe that we can ensure and develop a relative scale of measurement and can reduce the chances to a potential percentage as a value for understanding easily. Now, let's talk a bit about our microbiology house of the project. We discuss more on this on our project submission on the platform OmicsLogics. In the BioProject 821821, we realized through this dataset that the specificity of our cocktail is paramount. By using the disease status as a common ground, we found results that helped us make a case for the differences in gut microbiome population between male and female patients. In Bioproject 734525, it was specifically chosen for the variation in introduced through three distinct disease types: AD Alzheimer's, healthy, and minor cognitive impairment (MCI). We understand that through such differences exist in the microbial populace during different neurological disorders, differentiating them is all the more important and difficult. Summing up, in by Project 821 we conclude that the abundance of Ruminococci in AD patients can be a competent candidate to serve as an analyte. In the case of Bioproject 734525, it was pivotal SAE. During a literature survey, we came across an interesting paper that helped us conclude that we are on the right track. Through this work, we concluded that the firmicutes as a general indicator population as a positive test for Alzheimer's at a phyla level. In the case of bacterial family, Ruminococci won the jackpot. Coming to roadblocks to a solution. Number one, finding enough data to make a strong case for the feasibility of an early detection kit. Access to domestic labs to test our hypothesis, and finally identifying competent testing methodologies for our cocktail. Now let's talk about the business side of things. The team sincerely believes that this solution can only make sense when the purpose has a clear understanding of how to make it available to the consumers and make it a profitable enterprise in the healthcare industry. By conducting a very simple and rudimentary market analysis, we came across the dietary recommendation testing kit by Ombre, which was priced at $99. To make microbiome testing kits a viable option for healthcare professionals, we must make it affordable. To the best of our knowledge, we believe that these are the following expenditures we will incur as we go forward. Number one, hospital consultations. We believe that hospitals are an important facet of our stakeholders, and due to the proximity of the patients, they are in a better position to understand the needs of the patient. Developing a testing cocktail. This will involve the lion's share of our expenditure, for this will involve the heart of the product. Finally, B2B contracts to make excuse me, to make our product available through online vendors and microbiology labs for the consumers directly. Similarly, we expect our revenue to generate in the following ways. Number one, sale of the testing cocktail. We believe that we can make the testing cocktail compatible to an existing microbiome testing platform, hence our primary revenue source will be through the sale of a testing cocktail. Second, in the targeted sales. Hospital clinics that are in a position to treat neurological disorders or see a high influx of patients around the age of 40 can refer our product to their patients as a safety net. How will all this work? This will all work by using the Kanban method of manufacturing, where we manufacture our kits after an order has been placed. The reagents and chemicals will be made available to labs, which will also double as our warehouses. Following the order, the team present in these labs will begin assembling the pieces and follow strict quality control. Then the assembled ETK or the early detection kit will be picked up by the vendor and directly sold to the customer or the hospital. Excuse me. Our Recon series will be ranged from a $45 price tag to a $90 price tag. Our goal is to make this kit affordable to the middle to low income household by 2027. So, what is next for us? For our first bullet point, we believe identifying negative and positive tests for the identified bacterial family is important. Secondly, understanding the causal relationship between the gender and the chances of Alzheimer's. Thirdly, we have to finalize our literature review draft. And lastly, but not the least, for now we believe that reaching out to hospitals to understand the need for a solution and stakeholders holds similar importance. Our proposed timeline looks something like this. Come December and January, we believe that human practices will play a vital role. In February, we understand the logistics of our project. When we come to a chain of thoughts, we believed that we first understood there's an existing need for a detection kit. Secondly, we understood why existing kits are a concern for the healthcare industry. Third, we went out to identify specific populations of microbial biases through our pipelines. We did so by understanding two specific correlations between the gene-inducing Alzheimer's disease and the fecal microbiome. Secondly, the biomarkers of Alzheimer's disease and the gut microbiome. And in the end, what you see in front of you is a combination of our research in science and business as well. In terms of methodology, it is pretty straightforward. We understood that our datasets have to have two specific disease status, which means that they have to have a control or a disease patient status, which is what we exactly found. After running our pipelines, we analyze them with four specific key indicators as a philosophy. Number one being the abundant population in AD compared to control, less in AD and abundant in control, and similarly two other examples of how a similar population exists in both cases or they're absent in both cases. Finally, we understand further exploratory research is required to ascertain the practicality of the solution, the hypothetically it has shown promise. We understand that the required clinical testing to make it applicable for human diagnosis remains a huge variable in our solution's future. And finally, we understand the need for a robust supply chain to ensure that the manufacturing, storage, and the sales of this kit takes place smoothly. In our team, we have four passionate individuals: Ansh, Bhavya, myself, Nalaba, and Subhashri. We believe that we can change the world. Finally, at the end, we believe that our solution is very much similar to the role of a lighthouse. The solution is to warn users and allow them to change course in terms of one's habits and adopt a diet of supplements before it is too late. And with that, ladies and gentlemen, we come to the end of this video. I'd like to thank you so much for listening to me till the end and hope that we have made a convincing case for the solution proposed and the vision for making everyone's lives safer and better through microbiology. This has been Nilab Mukherjee from Team VIT Bhopal, signing off.